First of all, I would like to acknowledge Ngati Fatua and Ngati Manuhiri as mana whenua of the land we work on, to our funders, DOG and Save the Kiwi, through the Jobs for Nature and also Auckland Council, and a huge acknowledgement to our own team, the Forest Bridge Trust. Nothing of what I do could be done without the whole team effort. I've got a lot of slides, so I'm going to be quick. I might miss a few details, so please come to me if you want to know more. So, this one? Okay. So the vision, you've heard this before, I'm going to repeat it again. It's a connected landscape with healthy forests and flourishing indigenous wildlife from the Kaipara Harbor to the Pacific coast. So where exactly this bridge is, we have a map here, but I also have a map there. Two important reference points, Mataya on the left in yellow and Tawharanui on the right in yellow as well. And we have our central bridge, which is the shaded white area, and north and south flanks, which becomes our wider bridge. A bit of, which one? Okay. Background on this area, almost 60% is exotic grassland, 18% is exotic forest, and only 19% left of native forest. And this is what is shown in the map. So what is left, it's highly fragmented. We don't have huge national parks in this area, and they're mostly orange and red, which means critically endangered and endangered. So it doesn't look really good. So we really must protect these remaining fragments at all costs. Well, not all costs, but we have some pretty amazing fauna and flora. I completely ignore plants. Sorry to the plant people. I know there is amazing stuff out there, but I'm more of an animal person. So we have matuku, you know about it, it's critically endangered. We have long-tailed bats, also critically endangered. The lovely Hochstetter frog, which is at risk and declining in our area. And we have pateke, kaka, which are at risk but recovering, which is great. And of course, the star of the show, the kiwi. Kiwi are not officially threatened, but they are highly conservation dependent. So if we stop managing and conserving them, they'll be lost. Thank you. So a very quick history of kiwi in this area. This is where we are, on the arrow. Next one. Once upon a time, there was kiwi everywhere. With human settlement, they started to disappear. I'll go through because too many. Gradually disappear as we remove their habitat. And then we have, of course, the stoats that decided that kiwi chicks were really yummy and started eating them. And then the ferrets that can take adult kiwis. And of course, our lovely companion animals, cats and dogs, just to finish the job for us. So eventually, they did disappear from this area. We are not 100% sure when this happened. All I could find in the literature was this very short reference that says, reputedly heard by local residents around Tamahunga, the only certain records are of eggs found during scrap burning on our side of the Whangaripo Valley, that's Lee, in the 1960s, and one killed by a dog in the Matakana village in 1971. So this paper was from 76, so they were still around in the 70s, so probably gone around in the 80s, mid-80s. And in this just 3 lines, this a hint of what happened to them. It's basically habitat loss and dogs. So continuing with the story, the Tawharanui fence went up and predators were gone, and kiwi went back into this area in 2006. And then Mataya started its restoration project, and you're going to hear about Jill later. And they reintroduced kiwi in 2013. And they did great. So at this point is where Jill and Kevin thought, oh, how amazing it would be to have a whole corridor connecting these 2 populations. And this was a great dream, a great vision, and the Forest Bridge Trust was born. But we didn't really have the means or the resources to make this reality until Jobs for Nature and COVID. So we got that fund, and it's for 54,000 hectares under Mustelid control by June 2025. It's a 5-year project. And by Mustelid control, we mean 1 trap per 10 hectares and around 12 checks per year. So one of my first jobs was to look at habitat, Kiwi habitat and connectivity in our area. So I looked first at the central bridge, because this was the bridge we had in mind that they would use. So this shows plantations in red and native forest in green. But the experts said, nah, they actually really like pines. So this kind of became a really big fragment of Kiwi habitat. And this was great. There's a lot of Kiwi habitat in our area, and pretty good connectivity. So the central bridge made sense. But I also thought, well, wait a minute, the Kiwi don't really know about this central bridge. There's no real border or barriers. So I did another analysis that went sort of beyond that central bridge. You can see it in black, the central bridge. And this analysis looked at habitat and connectivity between these habitats, so it identifies corridors that they are more likely to use. So the green is the good corridors. The darker the green is the better, and the better chances of the birds actually following that path. So the central bridge, yes, it is a good corridor, but wait a minute, there's a really good corridor going down south. So that made us sort of rethink about this central bridge. It is a good starting point, but the birds will eventually use all this. It's more like a network. It's not one bridge, it's a real network of habitat. So we started rolling out traps, and this is sort of the starting point of the project. The areas that were already under Mustelid control, these are other projects, not just Forest Bridge Trust. So we have the Tamahunga Trappers in Tamahunga, Takatu Land Care in Takatu Peninsula, and Mataya and Atuanui, as we heard before with Heavy. They were already doing trapping there. So our first year was kind of slow. So this is January, and this is December. You don't really see much difference. That's because we were trying to recruit and scale up in the middle of COVID restrictions. So it was really hard to contact people, volunteers, landowners. So it was a really slow start, but it quickly took off. In the next 6 months, the whole Tamahunga buffer zone was built as preparation for the Kiwi translocation we heard earlier. And then since then, it's been sort of steady state, and we've been doing this count of areas under Mustelid control now. It's every 3 months. I'm going to go through them. So this is January, April, and this is where we are now. So we've come a long way. It's 32,000 hectares at the moment, and this is June. There's already a couple more thousands to add at the end of September. So you can see how this dream, it's actually coming true. So it's not just about throwing traps 1 per 10 hectares. It's a whole process of following up that traps are being checked to a high standard. I'm not sure if you can see the little flags there. These are catch of mustelids, stoats, whistles, and ferrets. This includes traps from other projects. This is all you can find on Trap and Set. But you can see that this is just the last 60 days. So all this pests been removed in the last 60 days. So it's pretty amazing how much we are taking out of this landscape. And as every ecologist would tell you, it's not what you're killing what's important, it's what's left behind. So that's why we measure these residual abundances. The problem with mustelids, and particularly stoats, is that they're really hard to detect. We know that tracking tunnels that have been used traditionally have very low detection rates. So you could have hundreds and hundreds of tracking tunnels and only one track, one detection. So we decided to try trail cameras at a large scale. We had 69 cameras in 3 main trapped areas, so the Kaibera, Tamahunga, and Takatu, and 42, and we were also keen to compare these with untrapped areas to see whether we could detect any differences. So we also had 42 cameras in untrapped areas. We consider 1 camera to roughly cover 100 hectares. And I tried to space them sort of evenly, but yeah, more or less evenly. So we lure the cameras with fresh rabbit and also a long-lasting lure. We use the Connovation resoakable meat flavor, which is this sort of orange thing there. They turn out to be pretty good. So this camera been out maybe 3 weeks, and you can see the stoat is clearly sniffing, still attracting the animals. We took 5-second videos, and we left them in the field for at least 3 weeks. That was the target, but it ended up being more like 1 or 2 months. And then we used the zip classifier, which is a little app. It's free on their website. It doesn't do AI, so you still have to go through your recordings manually, but it makes the work of collecting the data more easily. So you, in the black square, you will see your videos, and on your right, you have your list of animals, and you're just going through your videos and clicking, and then it will, once you're finished with your card, it will export the videos into different folders, so a folder for stoat, folder for possum, and it will also give you an Excel sheet with all the data, your camera, the time, the date, and the species. So it makes the work a lot easier. So there's no inputting data on Excel, which can be a real pain if you have hundreds and thousands of videos. So the results, a couple of cameras got stolen. Some of them had branches or cows interfering and had a blocked view. So we ended up with 97 cameras having enough data, so at least 3 weeks. And I'm going to present the results, sort of the raw results, which is occupancy, which is the percentage of cameras that had detections, and camera trap rate. So the difference between both, so the occupancy doesn't take into account whether it was 1 detection or 100, and it doesn't take into account whether the camera was a month out there or 2 months. So it's very basically where they are in the landscape. And the camera trap rate does take into account whether it was 1 detection or more, and it does take into account how long was the camera out there capturing this data. As you can see with stoats, overall it was really low. We only had 11 cameras with stoats. That's 11%. It's overall pretty low, as expected. We know that stoats occur naturally in very low abundances, and that's the problem. And then we found the opposite of what we were expecting. So slightly higher detections, occupancy in trapped areas than non-trapped areas, and same with the camera trap rate. So it's not a huge difference. It's not way higher, but slightly higher. So I don't want you to get the idea that the work we do does not make any difference. I'm pretty sure it's more about the method and how we don't really have the power to detect that difference yet. There's no national standards on how to do this, so this is very much work in progress. It's been a learning curve for us, for me. So we have the cameras, and we're going to maybe change it a little bit for next season and see if we have the good power to do that. In terms of the different areas, Kaipara and Takatu had sort of higher occupancy than Tamahunga. But then Takatu had the lowest camera trap rate. And there's also another thing that you see, some of the white triangles are really close, so it could have been the same animal triggering both cameras, and that would kind of inflate your positives. But then if we look at all the mustelids, so if we combine ferrets, stoats, and whistles, then we do see a difference between trapped and non-trapped areas. and that's more evident. So occupancy is pretty much doubled in non-trapped areas than trapped areas, and Crimea trap rate is 3 times higher. We only detected weasels and ferrets in non-trapped areas. We were very relieved to have just one ferret detection. We know how bad ferrets are for kiwi, so there was a big relief that it's not ferrets everywhere. And the other problem is that we had lots more cameras in trapped areas than non-trapped areas. And this was because we have a limited number of cameras, of course. And I was more interested to see what the abundance was in trapped areas than comparing with non-trapped areas. So I kind of get the idea, if maybe we had had more cameras down south, maybe we would have had more detections there. But overall, it was a really good try, I would say. I still have to look more at the data. I was still classifying videos on Monday. So this is all very new. I even haven't talked to this to my own team. So other species. So of course, this was targeted to mustelids, but we could see all the other species on camera. Possum, not very good news. 75% of cameras had possums, but really good news for Mataya. Well done. You can see a cluster of non-possum cameras there. Auckland Council also does possum control in Tamahunga and Takaturi, and maybe that's why there's a few more green triangles on those areas. And the non-trapped areas are just really bad, full of possum. Cats. Cats everywhere. You don't really see a pattern here. They're just everywhere. There's no really intensive cat control. We do some in some properties. We heard that Tamahunga does as well. But yeah, it's pretty bad. I think we have a problem here, and we should really be starting to think, how are we going to solve it? And then real nice bonus, kiwi. So we did pick up a few kiwi on the cameras in Takatu, Tamahunga, and Mataya. And also, if you can see this lonely dot completely outside of the areas where we were expecting them. That's Makaura. It was a really nice surprise. I had to check the location of the camera twice. I was like, is this really from this place? Is this not coming from somewhere else? But it was, and it was seen twice with a month apart. So we're pretty certain that, and it's an adult female. So I think that the bird is living there. It's not just passing through or just lost, which is really exciting. And it also, if you remember now, the connectivity analysis before is right where that deep south was going through. So it was a surprise, but not entirely unexpected. So continuing with kiwi. So we do kiwi monitoring. All the populations have their own monitoring. So Tawharanui does monitoring with Tosi and volunteers. Mataya does their own monitoring, and as we heard, Tamahunga does their own monitoring. So we were more interested to see how this population will be actually using this bridge. Are they expanding? Are we reaching that point where they are moving away from those translocated populations? So these are this year's results. I put 12 recorders in the Tawharanui buffer zone, sort of the immediate buffer zone, and the results were pretty good. 8 out of those recorders had kiwi calls. Some call rates are still on the lower side, but definitely there, so they were loud. So you could say very much there's a kiwi in this fragment. And the call rate in the bigger fragment, right close to the fence, it's pretty high. So a high density kiwi population is 5 calls per hour, and this is 4. So it's already a pretty good population there. So we knew from years, since the first translocation, kiwi had gone around the fence into the buffer zone, but it wasn't very clear whether it was just 1 or 2 or or there were more. I think now it's pretty clear that it's not just one or two. It's a proper population breeding there. We had quite a few duet calls, so they are breeding. And I also added here the kiwi that's seen on camera at the low kiwi here. And there was also a kiwi detected on a camera at the very top of Omaha Spit last year on the shorebird sanctuary there. And there's another, the other kiwi here on the left. It's a record on eBird. So it just paints a picture of, they are dispersing away from Tafra Nui. From our monitoring, there's 3 sites that we've been monitoring since 2021. So the 3 that are closer to the fence. And you can clearly see a trend there that is more and more calls. So I think if you think that the average juvenile dispersal is 5 to 6 kilometers, I mean, if you live in Tagatu, you're going to have kiwi the next couple of years, probably, hopefully. So we also help, I don't want to say we do it because I barely do anything here, monitor kiwi in the Mataya buffer zone. So we've had recorders the last couple of years. Most of them had nothing, but twice now we've picked up calls on the other side of State Highway 16. One of them had, I think, 20-something calls in one night. So yeah, they definitely crossed the road, which is really exciting. This was sort of the first sign that the Mataya kiwi population was expanding. But then everything happened really quickly, and in just 1 week, we had that kiwi on camera, which is the kiwi here at the bottom. And then one of our staff members, Paula, that lives in Tauhoa, said, "Wait a minute, I think we have kiwi prints on our place." And we're like, everyone that looked at the print was like, "Yeah, that looks like kiwi, but it can't be. It's so far. It's on the other side of the Hoteo River. How did they get there? There's a lot of farmland in between, not a lot of bush." So I had this with a big question mark, like, is this really kiwi? But then I was like, no, we need to know for sure. So I sent the picture to Rogan Colburn, who is the big kiwi expert, and he said, yeah, that's 100% kiwi. So if he says that, we're all gonna believe that. So yeah, definitely, and that's 9 kilometers north of Mataya. So yeah, if you live anywhere within 10 kilometers of Mataya, you might have kiwi already. Yeah, so this is really exciting, but it also stresses the need of how important this job is to go beyond your own population, and they're really great dispersers. So they can move long distances, as we saw before as well. So it's important that no one disregards it, like, oh no, they're too far. It can't be. We're safe here. No, they could be really pretty much anywhere. So we're going to move now from kiwi to our lovely bittern. We also do a little bit of bittern monitoring, mostly focused on Tafra Nui Peninsula. We do it mostly with recorders, but I also do a little bit of transect walks and see if I can hear anything. And yes, we do have bittern. They are breeding in that's the Whangatea Harbor. So these are recorders. We don't know exactly how many birds, because one recorder could pick up more than one bird. They are living in Christian Bay. We know that. That's been monitored for longer. And they used to be in Tafra Nui. There are records of bittern in Tafra Nui, but in the last couple of years, we've heard nothing, which is kind of puzzling. We can't blame predators. So it's definitely more to predators. So this year, I would love to go out with real people, not just recorders, so we can count them and have a better idea of how many they are. And I also want to expand the area that we monitor. So the broader Whangateo Harbor, hopefully a few places on the Kaipara side as well. We did a few beater monitoring at Mataya last year, and we didn't pick up any calls. But yeah, it's a huge harbor. I'm sure, I'm sure we'll find other places that they are living. We also do 5-minute bird counts. I'm gonna disappoint a few of you here. I'm not gonna present any results. This is more just to show you that we are doing it. It's around 65 stations. We monitor once a year. We go 3 times to each site, ideally. But 5-minute bird counts are really designed for long-term monitoring. There's no point in me showing you 1 or 2 years because you're not going to see any trends or anything. So it's more like I watch this space, and let's talk in 7 years. Because we have the equipment, which are these dog recorders that can also record bats, we thought, oh, we'll put them to good use and start looking for bats. A lot of people are really interested in bats. I think since the Bird of the Year campaign, we've had a lot of people thinking, oh, we think we've got bats. So for us, it's really good to be able to lend them a recorder and to see whether they have bats or not. This is at lovely Vara Bush. This was my first ever bat recorded. It was pretty exciting. So this is what a bat call looks like when you look at it. It's these pulses. They're very distinctive, so you can't really confuse it with something else. And we've been sort of lending out the recorders mostly to staff members at the moment, but yeah, we want to expand and do as much as possible. And so far we've had 3 100% sure bat, long-tail bats, and 1 potential. So what's Michelle? Yeah, you have bats in your place? It's been sitting on my desk and I thought, oh, I need to go through this. Yeah, so how am I doing with time? Fine. Okay, cool. So yeah, that's exciting, and we would really like to cover a larger area. We know that bats have very large home ranges. So yeah, and I've heard people saying they're in Mahurangi River, in Warkworth. I've heard them saying they're in Lee. So yeah, it would be great to have more sort of data to back all these observations and paint a clear picture, because we know they will benefit from all this mustelid control that we're all doing. And just to finish up, I want to talk about a little project we did with Friends of Awa Matakanakana. This is FOAM, a local catchment group that they do amazing water quality monitoring in these catchments. We applied to the EPA, that's Environmental Protection Authority, for some eDNA kits. Does everyone know what eDNA is? No? Yes? Well, eDNA is environmental DNA. So this is a freshwater tool. So you take a sample of water, and then you can sequence and look for DNA in the water, and pretty much everything that's gone or that is present in that stream will be shown in this analysis. So it's a really easy way. With just a water sample, you get a list of hundreds of species that are living on that waterway. And sometimes there's a few surprises in there. So we did this with FOAM. They did most of the work, so please, I just provided the kids and went to the field, but it was mostly them doing all the work. So we had 4 kits. So every sample point had 6 replicates. So you're pretty sure that you're picking up most of the animals that live in the stream. We sampled 2 catchments, the Glen Eden in yellow and the Matakana River in pink, I guess. And we had this sampling that we had the upper reach and the lower reach. And we wanted to see what's in there and what sort of differences there are. So what they give you back from the lab, it's a huge list of everything, from bacteria to algae, invertebrates, birds, fish, everything. So I'm not going to go through the list. We would be here for hours and hours. But some of the most interesting results are here. So the Glen Eden River had Hochstetter's frog in the lower reach, where you wouldn't expect them. But then you also pick up signals, so the frog could be up to 6 kilometers upstream, and the eDNA could still be picked up in the lower ridge. So we think there's definitely not a frog living there where we picked the sample, but it's more like coming from the upper ridges. Kakahi, which is the freshwater mussel, was detected in all samples. They are declining, so it's quite good that they are there. I know the problem with kakahi is they're very long-lived. They live like 50 or 60 years, which is crazy. So, and most populations are quite old. So people find this, the older shells or the older individuals, but it's not many juveniles being found. So they're still there, but maybe they're not really breeding there. So I think this, yeah, there's something to look for there. Long-fin eel and short-fin eel, both detected in all samples. Coda as well. The Glen Eden River, the lower ridge had inanga and redfin bully, and the upper ridge had a short jaw cockapoo, which is not very common, and it's vulnerable. That was really interesting. And banded cockapoo as well. And the Matacana River, so the lower ridge had kakahi, longfin eel, shortfin eel, coda, and redfin bully. But the upper ridge had more interesting stuff, some banded cockapoo, giant cockapoo, torrid fish, all at risk and declining. And so these wheels here are called the Wheel of Life. I don't expect you to read what's in there. I just thought they were really pretty, so I have them. And they also give you a score, a sort of a health score that they develop at the lab. And you can see, at least in the Matacana River, a big, well, not huge difference, but still good difference between lower ridge and upper ridge. As you would expect, the lower ridge is poor quality compared to the higher ridge. And then the Glen Eden had average quality on both samples, upper and lower ridges. So yeah, there's a lot of data in there, but I thought that I would talk about this because I think it's a really good tool. It's easy, it's not too expensive. With one sample, you can pick up lots of things. So if any of you are interested in doing this, I would totally encourage you to do it. And this goes into a national database. If you want to share your results, you can. If you want to keep them to yourself, you can as well. But it's great. You could go online and sort of explore all the results that they have from all the projects around New Zealand. And that gives you a really good indication of, where the species are. Yeah, it's a really cool project. Yeah, and that's me. I thought I would go over time, but I think I was talking too quickly. I finished. Thank you.